This is his favorite seminar. It's my favorite seminar. This is the one you all do. Those of you who are first years are going to watch these three and go, I can do that. We'll all chuckle. They're going to laugh maniacally, because they said the same thing. In 2019, this strange, short, bald guy showed up coming off a sabbatical. He did this little weird song and dance about, I'm back! And they're like, you weren't here before! And the upper guys were like, oh, look at him, he's relaxed. I don't like him that way. And yet here we are, 4 years later, and they're doing what you will do. You can do what they have done. You have to believe in yourself, and you have to believe in him. You can do those two things. Anything is possible through Christ. Honest to goodness. Right? You're going to enjoy today, because today you'll have three people who you know talking to you about work you can do for his glory. Right? Amanda, you're up first. Go for it.
Hi. For those of you that don't know me, my name is Amanda Imhoff. I am a senior biochemistry student here. I'm going to be talking about some research that I did over the summer. So I applied and was granted an RU experience, which is a research experience for undergrads at the University of Alabama at Birmingham. I worked with the Department of Biomedical Engineering to complete a project seeing how pulsatility affects von Willebrand factor. So to start, to give you guys a little bit of background before I dive right in this context, pulsatility is the rhythmic flow over the cells in the system. And von Willebrand factor is a factor within your blood that is responsible for the initial adherence of platelets in response to vessel damage or bleeding, in other words. So to start, I'm going to talk about different types of ventricular assist devices, or VADs as you'll hear me say. So a VAD will take over partially or completely the function of a failing heart. So if someone is waiting for a heart transplant or they're going through heart failure, they may need to be put on some type of VAD so then they can maintain a good quality of life. And so there are two different types of VADs that I'm going to be focusing on. There's a continuous flow VAD and a pulsatile flow VAD. And a continuous flow is used more often. It is smaller, it's more durable, it's cheaper. But one thing that happens with people with VADs, sometimes if they have a continuous flow VAD put into place, they can experience non-surgical related bleeding years down the line, 40, 50 years after implantation. And so we were curious if this had something to do with the type of flow within a continuous flow VAD, and if that affected the body's ability to clot blood through the von Willebrand factor. So that is what we were looking at while I was down there this past summer. And so these are two different waveforms. So a waveform is just, it characterizes the flow of the system. And so if you look on the bottom left, that is from a pulsatile flow VAD. And on the bottom right, that is continuous. What we want to see is more physiological, which is on the left. So in simple terms, pulsatile is more what you're looking for, and continuous is a little less normal to what your body would experience. And so to talk a little bit more about von Willebrand factor itself, the factor, when it is relaxed, it has a naturally raveled state. And so when it is raveled, that is when there's no flow over top of it. And so when there's blood flow that goes through your arteries or through your system, that unravels the von Willebrand factor. And when the factor is unraveled, there's a binding site that is now exposed. And that binding site is bound by an enzyme called ADAMTS13. And that enzyme is responsible for cleaving von Willebrand factor so that it can clot easier. However, if von Willebrand factor cannot return to its protected state or its naturally raveled state, then that binding site is always exposed. And if it is always exposed, then ADAMTS13 continues to cleave von Willebrand factor into smaller and smaller and smaller monomers until it can no longer be effective at what it's supposed to do. And so one of the benefits of having a pulsatile flow VAD is it allows your body to have relaxation periods where the von Willebrand factor can return to its protected raveled state, so its binding site is no longer exposed constantly. But in a continuous flow VAD, the shear forces in the blood flow is always leaving the binding site exposed. So we wanted to see if we could characterize that. So when I was down at UAD, I worked with a type of system called Microfluidics. And so that just allows us to look at the flow of a system on a very small scale. And so this is the process that I had to go through to make the devices. And we used a product called PDMS, which is a type of plastic. It allows for rigidity but also flexibility. So it is similar to what the cells would experience in the normal human system because you have some elasticity, but it's also not just, it also has some rigidity as well. And so we had to bind this system so then it was closed and there was no room for contamination or anything like that. And so once the device was bound and closed, it was then ready to go into the system after cells were put into the system. And so this is a system that we would put the device and the cells into. So this is called a perfusion loop. Think of it as like a mock circulatory system on a very small scale. You can do this with liters and liters of fluid, but we only did it with a few milliliters because that was just easier for us to do at the time. And so this is what our cells experience within this system. And the most important part that I'm going to be talking about further is number 6, the compliance syringe. And so one way that we manipulated this system to change the different waveforms, the characteristics of the flow throughout the system, was to change the volume of that compliance syringe that was open to the system. And so if we had the syringe open fully to 10 milliliters and had it open to the system, we got more continuous flow waveforms in this system. But if we closed this compliance syringe completely, we would get very similar to pulsatile waveforms. And so it was very interesting and cool to see that changing one small thing about the system gave us drastically different waveforms. And so we mainly used the compliance syringe to try and manipulate the system to put the cells through different types of conditions. And I characterized the difference in the syringe volume, comparing it to the average flow rate, the rate that the water or media is flowing through the system, and the pulsatility or the rhythmic flow through the system. And you can see that despite the fact that the first couple mils are a little bit of outliers, you can see that the majority of the data points are very similar. So if you recall from what I said earlier, closer to 10 mils is more continuous and closer to no mils is pulsatile. So despite there being very drastic changes in the waveforms that is produced, these cells are experiencing, for the majority of the time, very similar conditions. And so you can see that even though these waveforms are very different, they're experiencing the same average flow rate and the same pulsatile for the majority of the time. And scientists love that because that's one more thing that we don't have to worry about taking into account when we compare. So we could compare these cells because we know for a fact they are experiencing very similar flow rates and pulsatilities. So these are actually two waveforms that I was able to obtain from the system while I was down there. I have my continuous flow and my pulsatile flow. And you can see that with the continuous, there's very, very little movement. It ranges by only about 1.5 mils for the average flow rate. And so if you recall back to what I said about von Willebrand factor, when there's always flow, it's always unraveled, and it is always being bound. And so in this system, we would expect the von Willebrand factor to be cleaved continuously because it has some type of flow. But when you look at the pulsatile, that looks very similar to the waveform of what you would see in a physiological system. It has moments of no flow where the von Willebrand factor is able to ravel back up and protect its binding site. And if you look closely, you can also see that there is actually backflow or negative flow, which is similar to when your heart experiences diastole and refills. And so it has moments where it's actually refilling instead of always pumping out. And so in this system, the takeaway from this slide is that the continuous von Willebrand factor is always unraveled, and in the pulsatile, it has moments where it is able to go back to its relaxed state. So the cells that we use within our system were human aortic endothelial cells, or HAECs. And so these are the cells that you would experience, that would experience the system normally in a physiological system. And I was responsible for maintaining these cells, thawing them, growing them, maintaining their culture and things like that. And that was a really fun experience for me. And once these cells got to the proper level of growth, we put them into the devices that I mentioned earlier. And once they were in this device, we still had to wait to make sure that they grew and completely filled the microfluidic channel. And then after that, this is what it looked like. And so in this tiny little space, if you know what you're looking at, you can see that there are many, many cells within this little space. And so now this device in this system is now ready to be implemented into the perfusion loop. And so after we did that, we put them through the loop, and then this is how we imaged them with fluorescence microscopy. And so this is a type of, this is not what it looks like normally within your body. We don't have bright green or bright blue things within our cells. So this is just a way that we can image better. So the green is the von Willebrand factor, and the blue is the cell's nuclei. And so you can see that between the three different systems, there's a big difference between the von Willebrand factor that is present. One of the problems with this staining, though, is that we are looking only at von Willebrand factor. We don't know if it has been cleaved or if it has been bound or anything like that. We just see if it is present or not. And so we wanted to see if we could find a way where we could visualize the von Willebrand factor without it being cleaved or bound. So we found a way to introduce another enzyme into the system that was a competitive inhibitor for ADAM-TS13, which is the enzyme that is responsible for cleaving von Willebrand factor. And so if you look on the left, there is an A2 domain, which that is where ADAM-TS13 binds to von Willebrand factor. And so we implemented an enzyme into the system that would competitively inhibit ADAM-TS13 so it could not bind and cleave von Willebrand factor. And so our enzyme bound to the A2 domain, and so we were able to see von Willebrand factor without having to worry if it was being cleaved or bound or anything like that. And so that was a really cool experience to be able to reason my way through that with my mentor. And this is the imaging that we got. This imaging was not what we hoped for. Sometimes it doesn't always work out, but what ended up happening with our system is the shear forces that we were putting our cells under were too extreme, and the cells could not maintain their integrity, and they were coming off of the surface. And so we were only able to run these cells through the system for about 15 minutes when we were hoping for days. And so it's very hard to see any difference between the von Willebrand factor present, but this is something that can be continued obviously after I left. They can continue to work on this and find conditions that allow for the cells to remain in place so that we can run the system for a long period of time and actually see differences between the von Willebrand factor present. And one of the things that we're hoping for this research to come up eventually is to maybe find a way where we can implement a safer pulsatility that can be changed in patients with continuous flow baths that can hopefully prevent non-surgical related bleedings in the future. And so that is the long-term effects of this project, but I was only able to be there for once. But the National Science Foundation, they funded and rewarded my REU, but the people and the professors at Spring Arbor gave me the skills that I needed, and they helped me with the application process. And so a big thank you to everybody here that helped me with that. And yeah, so.
Hi guys, my name is Annika. Sorry, I'm a little bit more informal than them, but that's okay. So my name is Annika. The general purpose of my project was to create an undergraduate teaching laboratory. So a little bit more about who I am. I'm a senior. I'm getting a BS in biology with a concentration in cellular and molecular biology. I'm also on the women's reserve soccer team. And these are a few things that I love. Emphasis on dessert. Dessert is glorious. Yeah. Yes, sprinkles. Yes. So why should you do research? You're going to hear a lot about research today, but why should you think about doing it? First of all, it's fun. It is fun. Second, you can do things that are outside of the topics that you're learning in classes right now. So I'm in a bunch of classes right now, but my project has nothing to do with them, not even remotely. And in that sentence, it's like you can take a break from your school topics while doing this project, and it's lovely. Yes, you use those skills that you've learned in those classes, but the topics are different. There's some sort of discovery. You get to investigate a topic of your choosing. So my topic has already been discovered and gone through a bunch of times, but I made it my own, and I took that procedure and I made it so that we can use it here at Spring Arbor. And finally, trips. This is the Columbia River Valley in Oregon, where we got to go this summer. And Dr. Wyman took Jesse and I to the ASPB conference, which stands for the American Society of Plant Biologists. When we were there, it was very refreshing to have scientific conversations, because if any of you have friends that are outside of your major, it can be sometimes very exhausting to explain to them what you were doing without using exact scientific terms. I have a lot of non-scientific majors, and it's very, when I come home, they're like, "What'd you do in the lab today?" I'm like, "You wouldn't get it, but that's okay." So it was very fun to give what I gave to, or what I did, and it was very fun to see what they could give to me. So they give me ideas of what I could do, so they could give and take exchange. Some cons. It takes time and hard work, but everything does. So that's tough, but that's the reality. But what you put into it, you get out of it. So all of the hard work and time and effort that I put into my project, I'm now receiving the benefits, and I get to talk to you about this, and it's so fun. Personally, I love fruit a lot. If you didn't get from the mangoes before, my research is on mango peels. So every single time that I did a round of research, I used the peels, but then there's this fruit left over, and that's delicious. This topic is actually very interesting to me because I never really thought about fruit peels having microbes, or like having properties that could inhibit microbes, or anything around that topic. Bonus. Ethanolic extracted mango peels have been used to induce apoptosis in human cervical carcinoma HeLa cells. Super cool! That's crazy! A mango peel is inducing apoptosis in a cell in your body. What? Extracted bioactive compounds are also being used to make biodegradable food packaging films. I'm going to say that again. Biodegradable food packaging film. So like that plastic wrap is being infused with mango peels, and it's preventing microbial growth in your food, but it's also biodegradable. So in about 6 months, it completely degrades in the sunlight, which is awesome. Mango peels are also a huge source of biological waste. They produce about millions of tons a year, because if you think about it, it's just the peel, the fruit, and then the pit, and you only use the fruit. So about 50% of that is getting tossed in the trash, and it's attracting a lot of attention because of its antimicrobial properties. So what exactly did I do? This is the initial question posed of, do mango peels have antimicrobial properties? This is a very basic way of saying what I did, but I peeled the mango. I took as much of the flesh or the fruit off as possible, and then I would try to cut them up in smaller pieces, dry them in the oven upstairs. Once they are completely dry, which takes about 20 hours, I would crush them with a mortar and pestle, which is a very, very hard shoulder workout, in case you're wondering. Then I would separate these in half, like the total amount of crushed peel that I had. I would take that in half, put half of it in ethanol and half of it in methanol, so that I had a comparison of 2 organic solvents. Those would soak overnight in an orbital rotator, which essentially means they shake in a circle. Then I would filter them through Wattman filter paper number 1. Once that fruit extract goes through, you're almost done with the project. That's the extract I'm using. I would take the big chunks of peel that have been extracted, and those go in the trash. Then I would evaporate the liquid I just produced and filter through. Those go in beakers, and I would evaporate them under the chemical hood upstairs. After it had been completely evaporated, which by the way takes about 5 days, I would reconcentrate them with about 4 to 5 mils of the select organic solvent. And after that film is reconcentrated, I would cool them in -20 degrees Celsius in microfluidics tubes. So the actual assays I performed are zone inhibition assays. I will show you what they look like in a few minutes. On Mueller-Hinton agar plates. Mueller-Hinton agar plates are really simple plates where you can grow bacteria. And zone inhibition assays are essentially looking for cell death. So I put the extract into the media, it would diffuse into the plate and induce cell death. These are some of my results. These are the 4 bacteria I used and 1 fungus. And this is the labeling system I would use of my plates. So the top dot, or the top bowl, I guess I would call it, is the 100% mango extract in every single one of my pictures. Then you half each to the right, so it goes 100%, 50%, 25%, 12.5% of extract. The reason I'm doing that is to see the concentration differences in each of them. So the first one should be the biggest one because it's only a mango extract. And then hopefully the next one is about half that size, half that size, half that size, which you can see in most of these. Ethanol is my positive control, and then water is my negative control. This is the fungus plate, one of my personal favorites, just because it looks cool. As you can see, half the size, half the size, half the size. Beautiful plate. We are seeing tons of cell death. If you can tell the line, like almost a circle, within that is complete cell death. The mango had inhibited cell growth and essentially killed all the cells. And then this is our final one. A few next steps. Silver nanoparticles are, I'm hoping to synthesize them and then use them for biofilm inhibition and /disruption assays. And I'm currently working to optimize this procedure so Dr. Wyman can hopefully use it in microbiology. I would like to thank Dr. Wyman and all the professors who taught me all the skills I needed for this topic, and thank you for taking me to Oregon.
Just go in. Okay. Everyone hanging in there? Good? Okay, so I'm going to talk today about my summer at the University of Waterloo. That's in Canada, just so everyone knows. It's through a USRA grant, which is basically an REU for here, but it's through like the Canadian government. It's how like Canadian students potentially can do REUs in the summer. So yeah, who am I? Since I was doing research in Canada, it's kind of obvious I am from Canada, but I am Jessie, which I definitely didn't say at the beginning, but I'm a senior here, and I'm a biology major with a concentration in cellular and molecular biology. But at the beginning of my years here at Spring Arbor, I wasn't actually planning on going into cellular and molecular biology. I actually wasn't even planning on doing research at all. So I came in and I was looking to go into like the health career. I wanted to be a doctor, I wanted to go to surgery, all that crazy stuff. I had high hopes for that. But then I got here and I'm like, I really don't want to cut into people. And that was a really good realization to have before I continued on. And I hit my mid-freshman year and I was like, you know what? I really like the environment. I'm going to environmental sciences. And so I switched over to that concentration. And then over the summer, I was talking with Dr. Wyman about doing research, and he was really promoting the idea of doing research and working in the lab a little more, and I kind of realized that that was more the direction I was hoping to head. So if you decide to change your path a little bit here, it's okay. No one has a straight line, and if you do, that's great. But that wasn't my case, and I just wanted to kind of normalize that because it doesn't always have to be one straight path. I play on a softball team, and there's a little picture, but family is also super important to me, so I decided to put a picture up there because they really have shaped me to be who I am. And then that is a lab picture of Dr. Wyman, Annika, and I. Ashlyn and Abby are also in our lab, but we don't have a lab picture with them yet, so tough. So like Annika was saying earlier, we had the privilege of going to the ASPD conference in Portland. This was a really great opportunity to be able to present our research to people outside of the university as well as just to other science-related professors or like faculty. We were able to network a lot with different graduate students, with graduate programs and professors, as well as with different businesses, and that was just really really cool to be able to talk with people from all these different alleyways and be able to really learn more about the different alleyways that we could be taking after our undergraduate degree. So the things that I was looking at were the effect of algal allelopathic chemicals on the effect of those on cyanobacteria, as well as the effect of time on catalase function in Arabidopsis thaliana. Those were for my poster presentation, but I'm not going to talk about those today. If you have questions about them, feel free to come talk to me afterwards. So for my USRA, I did it at the University of Waterloo, which is shown at the bottom there. The science building is hiding in behind the tree, but that's where I was the majority of the summer. I worked with Dr. Kirsten Mueller, and in her lab, that's a picture of her up there, and she specialized in phycology, phylogeny, and taxonomy. So we did a lot of naming microbial species, and I'll get into that work once I start talking about my individual project. So what did I do over this summer? I did a lot of DNA extractions. Like some of you may have taken molecular genetics so far, and you do them a couple times, but I probably did 20 to 30 a week. And so it got to be, I got to be very comfortable with that procedure. I also looked a lot, I learned a lot about bioinformatics, so I was able to learn a lot of how to use different applications, as well as the coding that's involved in that. So I learned about Bash, which is basically like a coding language. And then I also worked with Chime, Genius, Mega, and then R, Mirlin, and these are all different applications that are used to analyze sequence data that you get and process that to be able to develop graphs and understand what is going on in your data. I also was privileged to be able to do field work over this summer, and this was just allowing me to be able to experience what it'd be like to be in a lab that you're allowed to do field work in. So it was the same techniques I would have used on my project, but I just went to a different location with different graduate students, and we were able to experience the collection of the data that we were then going to process, and that was really cool. We worked a little bit with qPCR, which is quantitative polymerase chain reactions, and so we didn't actually get to do that much with it because the graduate students are working on it now, but I kind of was able to be introduced to it at the very beginning, or the very end of the summer, because that's when they started working on it, which was a little unfortunate. But we got to do some of the trial runs while I was still there, and it was cool to be able to kind of understand how the machine works. And then finally, we did some gel electrophoresis, which is basically to visualize the DNA that we had just amplified through the PCR, because our data didn't transfer over to the data didn't transfer from the machine for our qPCR, so then we had to make sure that we got the correct bands and the correct DNA samples out of that. So my project was focusing on the Glenmore Reservoir, which is in Calgary, Alberta. This reservoir is like the main source of clean water in Calgary for their drinking water, for all sort of clean water purposes. And so what we were looking at is we were investigating microbial communities in the Glenmore Reservoir, and specifically we were looking for different microbial species, more so in cyanobacteria, of different microbes that have taste and odor or toxin releasing compounds, or are related to those species that do release taste and odor or toxins. So I was looking at the 2018 data from this, because it hadn't been analyzed yet, so the PhD student I was working with needed it for his thesis, and it was a perfect way for me to have my own individual project to build off of his thesis. So what we did was we extracted DNA from the filters that we got we vacuum filtered the water through the glass fiber filter that you can see there. And then we extracted the DNA from those filters. We sent them through 16S rRNA sequencing, which was actually still in Waterloo, so we basically just had to drop them off at the university, and then they were taken to do sequencing. And then we used all those bioinformatic techniques that I was talking about, all those applications, in order to analyze the microbial communities. And then just as an example, some of the toxins that could be used would be like microcystin, which is found in specific cyanobacterial species, and that's a very dangerous hepatotoxin, so it'll damage your liver. Thankfully, we did not find any species that could link to microcystin release, but we did find species that had geosmin release, which is a taste and odor compound that has like a muddy, earthy kind of taste. So you can imagine, like, if the Calgary community is drinking their water and it tastes earthy and muddy and not too good, that they're going to think that the water quality is bad, even though it's not actually dangerous in any way, it just doesn't taste good. So here are the two bar plots that we obtained from the sequencing data that we had. On the left is all the phylum that we found, or the major phylum that we found. So the most prevalent was the proteobacteria. It is really interesting to note, though, that there isn't enough cyanobacteria to be one of the main phyla that are represented. So it's actually in the less than 1% abundance, which is really good because most of the taste and odor and toxin releasing microbial species are found in cyanobacteria. But we still did analyze the cyanobacterial population that was found, and that is on the right. So you can see that there was no cyanobacterial species found in the May months, but then later on is when we started to see the appearance of cyanobacterial species. These plots are alpha and beta diversity plots. So on the left is your beta diversity, and that looks at the diversity between the samples that we were looking at. So the closer the spots are together, the more closely related the diversity is between the samples. So you can see that the majority of them are super close together. There's a couple that are pretty far away, but for that big clump, all of those samples have a very similar diversity between them. And then on the right is the alpha diversity plot, which is the diversity within the sample itself. So the higher up it is on that graph, the more diverse the sample is within itself. So the mid-lake samples, which is just a location in the Glenmore Reservoir, had a much more consistent high diversity through all the sampling dates, whereas the Weasel had just another location in the Glenmore Reservoir, was a little bit more widespread and had more of a variation in diversity throughout the summer. And then finally, this is the phylogenetic tree. I know it's very hard to read. It was just a really big plot, so it didn't quite fit very well, but I can explain what's going on, so don't worry. This is the phylogenetic tree that we produce through Mega and Genius. And so what we had to do with that is we took the sequences that were initially identified as cyanobacterial species, and we put those through BLAST to see which ones would align with specific species of cyanobacteria or what alignments we would get. And we took the sequences that we got from BLAST and we aligned them in Mega and Genius. So that's basically putting them all in the same region of DNA so that they can all be in order and be able to be related. And once we did that, we were able to form a phylogenetic tree from that, and that showed us that a lot of the ASVs, which are kind of the shorter names, they're numbered, those are the different sequences that were from our data, and those aligned a lot with Synechococcus species, which is a species that is a known geosmin producer. So the majority of the sequences that we found were related to known geosmin producers. And then another one is Nautilinea, which is kind of closer to the front there, and that also relates to a couple of the ASVs, the sequences, which is also a known geosmin producer. So I would like to thank the University of Waterloo as well as Dr. Mueller and her lab. I worked a lot alongside Timothy Charlow, which is the PhD student that really taught me basically all the techniques that I was using throughout the summer. I wanted to thank NSERC, which is just the research council that awarded the USRA. And I'd also like to thank Dr. Lyman for the mentoring that he's provided and the drive and push that he's given sometimes when needed, in order to give us the opportunity to have these research experiences. And then just thank Spring Arbor just because of all of you professors who have provided the information and the experience to get here. So yes, and these are my references. Yeah.
One for you and one for you.
Okay.
So Amanda, if the pulsatile is pushing forward and back, the back is also going to tend to unwind the von Willebrand factor too, isn't it?
Because it was more of a passive movement, we didn't feel like we needed to take that into account because of it being just so, like the force behind it was so minimal.
The other question I had was the continuous flow. The units on there looked like the pressure of the flow was pretty low. Is it that easy to I didn't think it was that easy to unwind. Von Willebrand factor.
The way for like in the continuous system is that like because of it being constant, it's not. You're right. You're correct. It's not easy to unravel von Willebrand factor, but because of the fact that the pressure is constant, it kind of like we thought of it as like, kind of like chipping it away until it opened, and then once it was unraveled, it remained that way.
Okay. Because von Willebrand factor disease maps to certain alleles of some are in Adam TS13, others are in von Willebrand factor. Did you look at any alleles
We did not.
of von Willebrand? Is anyone going to do that?
They might when we kind of figure out the system a little bit better.
Okay.
They were mentioning that would be a nice step.
Okay. Yeah. Andi, why did you do zones by, was it a volume thing? Instead of taking out the bits in the auger, how come you didn't use discs?
Discs actually didn't work. It was.
There's not enough stuff on the disc?
I think so. It also, whenever I did, like my positive control didn't have any cell death. So I personally believe that all of the ethanol or like organic solvent, I think there wasn't enough there and it just evaporated. And the ethanol and methanol, so sorry, was like the way that it diffused into the plate. So it had no way to move. Or like, yeah.
You didn't do tube MIC sort of tests? Minimal inhibitory concentration tests? No. That'd be kind of the next thing you do, I guess.
Yeah.
Solvent's going to be a big issue, though. But that would be better control for solvent.
I just didn't see any cell death when I put discs on top of the media, but
Yeah,
I
your
did.
concentration's probably too low, I guess.
Amanda, your syringe that you were using, is that just acting like a reservoir? Or
Yeah.
That's all it was doing?
Yeah, it was. It was just another way to either add resistance to the system or take it away.
Oh, darn.
Yeah. The perfusion loop system was much simpler than I was anticipating it would be. It's very straightforward, and we could even make it here.
I'm a little curious if that's the case, maybe why you had it vertical rather than horizontal.
We tried it horizontal, and it just kind of threw the waveforms.
So that's why, yeah.
Yeah.
Perfect.
Yeah.
Hennigenberg,
What's up?
your silver nanoparticles, is that something that Dr. Wyman just mentioned to you, or is it if you know a little bit about how you might be making those? I'm asking because I'm wondering if I've found a way to make them pretty easily.
I've made them. It was actually fairly easy synthesized process.
Okay,
For synthesis.
good. How do you know you made it? Is there any like going on? Or?
Yeah, so the color change is what tells you that. So it essentially starts off with like an orange, like a clear solution, and it turns to like a murky brown.
see that water source, is that stratified?
Yes.
That's why you have bacteroids? Because
Yeah.
it gets bottom and it gets anaerobic?
Mhm.
Okay, when I saw the bacteroids up there, I was thinking, what?
Yes.
If it's aerobic. Second question, the proteobacteria, that's mainly enterics. Do you have sewage feed going into that water source?
There's not supposed to be. That's why it was.
That's what I was questioning. When
Yeah,
enterics are that high, that suggests sewage contamination.
So there is glacial runoff from the location, so we're not sure if that could have something to do with it, but there shouldn't be any sewage in it because it is the drinking water source for Calgary.
So there aren't any golf courses or anything near there,
No.
so you have phosphate runoff from.
No,
Okay.
because that's the worry is that like they're really focused. The purpose of it is to they're very focused on avoiding algal blooms, which is why they're following the microbe
Right.
population a lot.
I'm not familiar with Clear Lake in California,
No, I'm not.
which you can literally dip your hand in the water and there's aphanzomina, which is a microcystis maker
Yeah.
in the water. It's terrible.
That's
Clear Lake
terrifying.
is so clear.
Yeah.
Give me one more hand, please. I want to emphasize again, for anyone who isn't a senior, and by the way, seniors, congratulations, keep it going. The rest of it, literally, you can do what these three ladies just did. It may seem unimaginable, unattainable, unreverable. Trust me, it is scientific. The reality is, you can do this. One of the great joys I get is watching you do the thing you didn't think you could do. Because all the faculty did the same thing when you were your age. Some of us had hair then, possibly even goals. Thank you. But the reality is, we were all you. You can do the thing you find hardest to believe. I know where this is. Some of you do. Tom does. This is the Dean Dome in North Carolina. I had the distinct pleasure of watching many a basketball game there. And all of these are the folks who made All-American. In effect, this is what our All-Americans do. These are our All-Americans. Odd man in many ways. But I got this interesting idea that this is my Dean Dome.
Yeah.
This is my Dean Dome.
Yeah.
I love being in this space because I get you in your first year, and I get to watch you go every year after that. But I start with you here. And I would love to see something like this on these walls. And so I'm going to ask you all to help me fill this space with the work that these young people do and you are all going to do. Because all of you can be All-Americans. Give me 5, 6, 10. You can all be All-Americans. Please help me do that. It's that important to me. Thank you again for coming today. Really appreciate it. If y'all have questions, ask the ladies. Otherwise, enjoy the afternoon.